circuits that we've introduced into citrus plants. So these are transgenic citrus plants. And on the right side of the slide, you can see a Hamlin transgenic with one of these constructs in it. After we've treated it with Rebus, they start to flower, and then they produce fruit. So that, in a nutshell, summarizes the broad strokes of what I'm going to be talking about. Okay, so this project started with conversation with my colleague Mike Bruce at UCR, who, as I'm sure many of you know, is a distinguished citrus geneticist and a breeder responsible for many varieties. And he impressed upon me a point that was made very nicely this morning by Danielle Seymour, that the juvenility of citrus creates this real problem for genetics. Because, as we were told earlier, this time from when the seed germinates to when it flowers and produces the next generation that you would use in genetics can be several years. It varies by variety, but it's a very lengthy process. And so a lot of the techniques that are used in traditional breeding to move disease resistance genes into organisms are essentially, they're very difficult or slow in citrus because of this biological limitation, or this biological feature of citrus and many other tree species. So just as one example, Chandrika Ramadagoo, who's another colleague at UCR, who we're collaborating on some of this work. She's extensively characterized HLB resistance and tolerance in a large number of citrus varieties and citrus relatives. And she's observed, for example, that Australian relatives, like Aramo citrus shown in that picture, are quite resistant to HLB. And she's done crosses with those varieties and observed that the F1s, so the progeny produced from those, are also resistant to HLB. The problem is that it's a slow process to move that resistance gene, or the underlying genes responsible for resistance, into varieties that are going to look more like a commercial variety, because these hybrids still have a lot of undesirable traits from their Australian relatives. So she's very actively working on that. It's traditional breeding and is making a lot of progress. But our idea that we tested with the help from the CRB with funding was to see if we could develop methods to try and speed this up using genetic engineering. Okay, and the approach we took is fairly straightforward and builds on years of work in many plant species understanding how flowering is regulated. And I won't go into all of the details. The key point is that there is one protein in particular called flowering locus T, or FT for short, and it is a master regulator of flowering. When the right conditions are experienced by plants, which could be day length depending on the species, or temperature, as it appears to be the case in citrus, this gene gets turned on, its protein gets made, and it moves into the shoot apical meristems and instructs them to develop flowers. And so we've had this knowledge for a very long time, and people have been trying to use FT to mess around with flowering time for some years. In citrus, there was a paper published in 2005 from Endo and collaborators that put FT into transgenic rhizome. They used what's called a constitutive promoter. So that means that the FT gene that they put into the transgenic plant is always on. And they saw that indeed they could get juvenile transgenic citrus to flower quickly within months of generating the transgenic plants. But what they observed was that there's a relationship between sort of the health of the plants and how much of the gene they express. So when they make a lot of FT, they're dwarf and they have all sorts of defects in their architecture that makes it difficult to work with them. And the problem is that normally this gene isn't on all the time. in the way that they set up this strain, it comes on at specific times and in a burst to trigger flowering. And so really the FT part of this has been known for a long time, and our piece technologically is to develop a chemical switch that allows us to turn genes on at will so that we can keep them in an off state and avoid some of these problems that come from high-level overproduction of genes like FT. And there's another tweak to this that I already mentioned. When we developed this technology, we really wanted to make sure that we could do this effectively in crop plants. A lot of the technologies for turning genes on that had been developed prior to our work use chemicals with toxicity, or they're expensive, and they wouldn't really be well suited outside of the laboratory environment. So the switch that we built is turned on by Rebus. The generic name for that is mandipropamid. The other logic to using an agrochemical as the controller of the system is that agrochemicals have been optimized for UV stability and uptake and things like that. So they generally have gone through a lot of testing to be optimized. And indeed, we find that that Mandy works very effectively in the system that we've built. I'll call it Mandy for short. So FT is the gene that we're trying to overexpress, and the switch that we built is controlled by this agrochemical Mandy, or Rebus. Okay, so there's two parts to this work. One part is the whole protein engineering piece, which is how did we build a switch where we can turn genes on and off by spraying the plant with an agrochemical. And then there's the piece which is using this for an application, the application being accelerating generation times in citrus. So my talk, the remainder of the talk will be both of those, but I will try to go through the protein engineering part rather quickly because I know that it's the citrus part that most of you came here to hear. Okay, so as Melinda mentioned, my lab, the majority of my lab studies this stress hormone called ABA, abscisic acid, and many years ago, we figured out, my group and other groups figured out how this hormone is able to do what it does in plants. And to make a long story short, there is a receptor, there's many receptors, but for the purposes of this talk, we'll just say there's one. And that receptor binds to the hormone when the hormone is made after the plants are stressed. That causes the receptor to form a protein complex with another protein and inhibit its activity. And the main point of this is that this mechanism where the chemical directs two proteins to come together and stick together is given the name chemically induced dimerization. You don't really need to track what that means. The bottom line is it means that this signaling module is very well positioned to build genetic circuits where we can turn genes on and off at will. And about a decade ago, a lab at Stanford showed that they could create a system where ABA turns on gene expression in mammalian cells by fusing the receptor to a protein that will bind to DNA, and the other part of the module that's called HAB1 to a protein that is going to help activate transcription. To do work in plants, we cannot use ABA as the controlling ligand because they're actually making it, and what we ultimately want is to gain control of the transcription. So as I mentioned, we set out to create a switch that we could control with Rebus. And there's sort of two iterations and steps to this project. It started showing that we were able to change the receptor so that it could be activated by mandipropamid or Rebus. And in the original iteration, what we showed was that once activated, the modified receptor would turn on the signaling pathway that it normally regulates, which is this stress tolerance thing. what we then set out to do was to try and convert that idea into a sort of generic module that we could use to control gene expression in plants using an agrochemical. And again, I won't go through all the details, I'll just stay at kind of high level. But what we really had to do in this protein engineering project was kind of rewire the pieces so that they wouldn't talk to the native plant signaling pathway that we didn't want to activate, but could talk to one another and be used to create a genetic circuit. And in the lingo of synthetic biology, that's called building an orthogonal signaling system. But that name is not really important. The key point is that we had to change this so that it could work without turning on ADA signaling. Because we don't want to activate ADA signaling, we want to turn on FT. The details of this are not critical. We succeeded in building this module and a crystal structure determined by a collaborator of mine named Francis Peterson at the Medical College of Wisconsin explained very nicely how this system works. Basically, there are little knobs and holes put on the proteins that allow the modified proteins to interact with one another, but they can't interact with the normal proteins that they would usually bind to. And so that creates this insulated switch that we can use to build the type of transcriptional circuit that I mentioned to you earlier. And when I say transcriptional circuit, I'm really meaning a piece of DNA where we can have these proteins present in the cell. We're going to treat them with the inducer, mandiprocamid, and then the gene that we're trying to express, FT, is going to come on. And in the lingo, those are frequently called transcriptional circuits. But it's really just a genetic switch to turn the gene that you're interested in on and off. Okay, so as I'm sure you're aware, working in citrus takes some time. It's slow, and we had a learning curve. So the first experiments we did used this switch in Arabidopsis, and I won't go into the details. The bottom line is that we created a piece of DNA that could be introduced into plants using agrobacterium-mediated transformation, and we're able to insert into there whatever gene it is we want to turn on and off. And the pictures of the plants turning green at the bottom are indicating that within 24 hours of treating Arabidopsis plants that have been engineered with this system, we're able to turn gene expression on. And it works with plants grown in soil, and it seems to work at very low concentrations as well. Okay, so the switch worked in Arabidopsis, and this is where we started moving into citrus. The first thing we needed to do was to put the pieces together so that we could turn on that flowering time gene, FT, when we treated the plants with this chemical, mandiprocamid. That is fortunately not too hard to do. I want to point out that there's, on the piece of DNA, what's shown there is the DNA that gets moved into the plant and inserted into the plant's genome. And it contains all the necessary components to respond to that chemical and to turn the gene on. On that piece of DNA is, on the right-hand side, is a gene called RFP for red fluorescent protein, and that will just be relevant later. It allows us to see the transgenic plants. They basically glow under the right wavelengths of illumination. And we call this construct that we built in the system IFT for inducible FT, and that'll be on some of the slides. Okay, so we started off first putting this into chorizo because chorizo is really one of the easiest varieties to make transgenic. In citrus, it's a citronge, so it's important as a rootstock, but maybe not as a sign, but obviously not as a sign. We generated several chorizo transgenics with this IFT DNA in them. On the left-hand side there, we're showing one transgenic that has been cloned, and the leftmost plant is genetically identical to the right one, but the left clone was not treated with mandiprocamid, the inducer, and the right one was. And you can see that these young juvenile citronges are flowering when we treat with the chemical. We've had those plants, the Carrizo transgenics, are the most advanced and the ones that we have the most experience with. But in the last year, we've been moving this into a much larger number of varieties that I'll say a little bit more about in a couple slides. And on the right, we're showing data from transgenic Hamlin. Again, treatment of these transgenics with the inducer triggers flowering. And the middle panel shows that we're measuring how much of that FT gene is being produced when we treat with inducer or a control. And you can see the orange bars are much higher than the blue bars. We're getting, depending on the transgenic in the experiment, about 10 to 40-fold induction of this gene, and that is sufficient to trigger flowering in the transgenics. Okay, so I think these two slides illustrate that the system is working, but I think there's a little more to do before this is going to be really useful as a breeding tool. Let me make one point. Got ahead of myself. So this picture on the left are of our transgenic Carrizo, one of the first plants that we generated. And you can it might not be totally clear from this image, but they have a normal growth habit. They're very productive. We're getting lots of fruit off of them, at least for greenhouse-grown plants. And so we really think that this is having minimal effects on the development, unlike those earlier strategies where plants were dwarfed and had other abnormalities. So I think that's a good sign in terms of using this as a genetic tool. But the more important point that I wanted to make was that we've recently demonstrated that we can use the pollen from these plants in crosses. So it would be kind of useless if there was some problem with the pollen in these plants, because then we would, we could make them flower, but we wouldn't be able to use them in crosses. And so we've used some of those Carrizo lines in different genetic crosses. The left cross there shows seeds that are embryos dissected from their seed coats under the right wavelength to make the seeds glow. And those are progeny, or the offspring from a cross of a Meyer-Lemon female with the Carrizo transgenic. And you can see that there's red seeds there. That means that the foreign DNA that's present in that Carrizo plant got transmitted into the progeny, or the children seeds there. And we've also recently demonstrated this in a cross of a sibling line to Siamese sweet pomelo. So we think that this checks a pretty important box. Perhaps it's not too surprising, but we really wanted to have this information because obviously to use this as a breeding tool, that's pretty critical. Okay, so in the last year, we were funded to start asking the question, how broadly can this technology be used in citrus? And we've introduced the construct into a number of varieties. There are some additional ones that aren't shown on that table that we just don't have data for yet. And what you can see is the pluses indicate that we were able to trigger flowering in the transgenic plants that we generated, or at least one of the transgenic plants that we generated. And so we validated that it works in sweet oranges, mandarins, at least W. Urquhart, lemon, and then a couple rootstock varieties. So far, we have not been able to get it to work in C35 or other trifoliate, pure trifoliate rootstock varieties. We're not sure if there's some problem with those genotypes that's just going to preclude it from working, or if it's a problem with the transgenesis. We'll sort that out. But the takeaway from this is that most of the varieties that we put this DNA into are working as expected and flowering after we treat them. Okay, so just to summarize, we built this little switch that allows you to turn a gene on in a transgenic plant with an agrochemical. We used that to create a genetic construct or piece of DNA where FT would be turned on by that agrochemical. We put it into several different citrus varieties, and we're able to show that we can induce flowering in juvenile citrus, and then pollen that they produce can be used in crosses, and we're also able to get fruit from those transgenics. In about, I would say, going from the time we planted the seed to the time we harvested the fruit from those transgenics, it's on the order of a year. So that's kind of a big speed up over the normal timelines for going from seed to seed. Okay, so yeah, I think that where this project is going then is towards introducing this into lots of varieties and starting to use it to try and speed up these genetic experiments that are ongoing, for example, to introduce resistance genes for HLB, or to introgress them from wild relatives. So I thought before I finish, I would put just a couple discussion points up. So in order to make these plants flower, we had to make a transgenic plant. We had to put that foreign DNA into the plant. That makes those plants technically genetically modified organisms, and as I'm sure you know, GMOs, as they're called, are regulated by numerous regulatory bodies, both in the United States and abroad. But in the US, and I should stress this is particularly the case for the US, the USDA decided that as long as your product, so let's say we were to use this in a breeding program and we got something that was really exciting or interesting, as long as we get rid of that foreign DNA, the USDA does not consider it GMO. So we could use this to speed up many cycles of breeding, and then in the very last step, get rid of the foreign DNA. And by US law, that would be fine, and you could sell that. The picture is different in Europe, and so that means that this approach, until the regulatory framework gets worked out, is always going to have some caveats about how widely used would any varieties built with this be. But I do think that it is definitely worth pushing forward on, if only just for research purposes, to help speed up the genetic identification of the causal genes underlying those kinds of traits like HLB resistance. Okay, so I'll just end with this one last point. I'm just I guess, highlighting a recently funded grant for Chandrika. This is a conventional breeding project. I mentioned already that she has F1 hybrids that are resistant to HLB. She has more advanced hybrids now that have been back-crossed. And she's the lead on this project to try and test and improve these varieties. What we're trying to do is use this ability to trigger flowering and generate fruit in juvenile plants as a way to evaluate fruit quality, or at least to help separate hybrids that have zero chance of being interesting from those that might be interesting. In that approach, it's sort of like the transgenesis is a side route, and the products that Chandrika develops, the varieties would never have had any foreign DNA in them, and they wouldn't be subject to the types of regulations that I was alluding to. Okay. So, just to acknowledge, the majority of the protein engineering and citrus work was done by Songwell Park. I've mentioned my collaborators already, and I'm very grateful for the funding from the Citrus Research Board, and I'm happy to take questions.
Do you have any questions from the audience? One in the back.
If you were to graft a non-transgenic scion onto transgenic rootstock, would you be able to induce flowering?
Wonderful question. Oh my gosh, I fully anticipated that. I did not plan that question, just FYI. Okay, so this has been one of the great disappointments of this project. What you're referring to is the ability of FT to move throughout the plant and trigger flowering. So historically it was identified and called florogen, and we now know that florogen is really essentially this protein moving throughout the plant. And in many species, if you do the experiment you described, you have a rootstock that makes lots of FT and a scion that has normal levels of FT. The FT will move from the roots or the lower parts into the upper parts and trigger flowering. And so we had hoped in the beginning that this would be the case in citrus. And the short answer to your question is we have tried pretty hard to try and get that working. This is just the cartoon there is to illustrate, like a chorizo trifoliate making lots of FT and moving into, say, a mandarin scion and triggering flowering. As in spite of how hard we've tried, we haven't been able to demonstrate that. A lot of the experiments that were done to show those types of effects were done with pretty young seedlings in tomato and Arabidopsis. So we're not giving up on that idea, but the simple idea of having like a rootstock that induces flowering on whatever variety you want does not seem too likely at this point. Thank you for that question.
Okay, any other questions from the group? With that, thank you guys. Thank you once again, Sean.